Med students on the rise, reaching for the skies. Diagnosing symptoms, we the wise guys. White coats on our backs, knowledge in our brains. In the world of medicine, where we break chains. Clinical rotations.
And welcome to the first episode of Basic Embryology Made Simple. Today's topic is going to be about the primordial germ cells and the development of the gonads. The primordial germ cells derive from the cells of the epiblast layer during the second week of the embryonic development. The BMP4 from the trophoblast induces the germline specific genes in the epiblast. Between the 4th and the 6th weeks, the primordial germ cells migrate via amoeboid movement from the yolk sac to the wall of the gut tube, and then through the gut mesentery to the dorsal wall of the body, and then alongside the dorsal body wall to the genital region, which is gonna form the future gonads. During their path, the primordial germ cells continue proliferating by mitosis during migration. Some primordial germ cells may get misplaced or detached from their path, and they reside in the extra gonadal sites. They continue to proliferate within these regions, and this will lead to the formation of a tissue that is known as teratomas, which is a tumor derived from the germ cells. The reproductive system actually develops in close integration with the primitive urinary organs, since they share a common tubular structure for uresis and gamete transfer. The intermediate mesoderm gives rise to ridges on both sides of the dorsal body wall known as urogenital ridge. This urogenital ridge has two parts, and I made a simple sketch so that we can discuss it. It has a longer, much larger lateral portion called the nephrogenic portion, or the mesonephros, which is relative to the urinary tract development, and a smaller, more medial portion called the gonadal ridge, or the gonadal portion of the urogenital ridge, which will give rise to the gonads. So again, as we mentioned, by the 6th week, the germ cell migrate from the yolk sac to the mesenchyme of the dorsal body wall, and by their arrival, the germ cell induce the 𝕿𝖆𝖌𝖘 epithelium at the level of the 10th thoracic segment to generate somatic support cells. The proliferation of adjacent somatic cells creates swelling, which will give rise to the primitive gonads. The somatic support cell encapsule and nourish the primordial germ cells, and they give rise to the ovarian follicles in the ovary, and they give rise to Sertoli cells lining the seminiferous tubules in testes. The genotype of the embryo is developed or established at the fertilization. So if we have a spermatozoa that has an Y chromosome, so the zygote will have XY and you have a male. If the spermatozoa have an X chromosome, we will have a zygote that has double X chromosomes, and it develops as a female. During the first 6 weeks, the embryo remains in a sexually indifferent or undifferentiated stage. This means that genetically female embryos and genetically male embryos are phenotypically indistinguishable. During the 7th week, the indifferent embryo begins phenotypic sexual differentiation. Let's take a male embryo, for example. The primary sex course develop from the gonadal ridge and incorporate primordial germ cells, which migrate into the gonad from the wall of the yolk sac. The primary sex cord extend into the medulla of the gonad, and they lose their connection with the surface epithelium as the thick tunica albuginea forms. Starting at the end of the 6th week, the SRY gene on the Y chromosome is expressed in somatic support cells. This initiates a developmental cascade for male reproductive structure to start to develop from the somatic support cells. Without this, the SRY gene expression, the embryo develops by default as a female. The SRY enables the somatic support cells to become precursor cells by the release of the TDF. TDF stands for testis determining factor. This factor will induce the formation of the male gonad, and in the absence of this TDF, the genital ridge will develop as a female gonad, namely the ovaries. The precursor cells recruits mesenchymal cells to form Leydig cell and testicular endothelial cells. Once established, testosterone and dihydrotestosterone, which is a derivative of the testosterone, are secreted to further assist in male gonad genesis. The testosterone induces the differentiation of the mesonephric or the Wolffian duct into ductus deferens, epididymis, and the seminal vesicle, while the dihydrotestosterone induces the differentiation of the urethral epithelium to prostate, the genital swellings into scrotum, and the genital tubercle into penis. Sertoli cells secretes androgen binding factors which will enhance the effect of the testosterone and the testosterone from one part, and they will help them to further induce differentiation of cellular differentiation into male gonads. Also, they release the Mullerian inhibition factor, or also known as the anti-Mullerian hormone, which will lead to the degenerescence of the paramesonephric, or also known as the Mullerian duct. The testes initially develop within the abdominal cavity. They later descend into the scrotum due to disproportionate growth of the upper abdomen away from the pelvis region. This is called the relative descent. The gubernaculum is a band of fibrous tissue that extend from the caudal pole of the testes to the scrotum along the posterior abdominal wall, and it is thought to play a role in guiding the descent of the testes. And the adult main ruminant of the gubernaculum serve to anchor the testes within the scrotum. The primary sex cords in females develop from the gonadal ridge and incorporate the primordial germ cells, which migrate into the gonads from the walls of the yolk sac. The primary sex cord extend into the medulla and develop into 𝕿𝖆𝖌𝖘, which eventually degenerate. Later, secondary sex cord develop and incorporate the primordial germ cells as thin tunica alba-genia forms, same as it happens with males. The secondary sex cord break apart and from the isolated cell cluster called primordial follicles, which contain primary oocytes and surrounded by the layer of simple squamous cells. The primary oocytes are derived from the primordial germ cells, where the simple squamous cells of the primordial follicle and the ovarian connective tissue stroma are derived from the mesoderm. The ovaries originally develop within the abdomen but later undergo relative descent into the pelvis as a result of disproportionate growth of the upper abdomen region away from the pelvic region, same as the testes. And they descend by the intermediate of the gubernaculum as well. But the gubernaculum actually gives some derivatives, which are the ovarian ligament and the round ligament of the uterus. Also, the peritoneum evaginate alongside the gubernaculum to form processus vaginalis, which is obliterated in the female later in development. The para-mesonephric duct developed as mesodermal invagination of the lateral surface of the urogenital ridge. The cranial portion of the para-mesonephric duct developed into the uterine tubes. And the caudal portion fuse in the midline to form the 𝕿𝖆𝖌𝖘 primordium and thereby bring together two portional folds called the broad ligament. The 𝕿𝖆𝖌𝖘 primordium then develops into the uterus, cervix, superior 1/3 of the vagina. The para-mesonephric duct project into the dorsal wall of the cloaca and induce the formation of the senovaginal bulb. The mesonephric or Wolffian duct and tubules develop into female as part of the urinary system because these ducts are critical in the formation of the definitive metanephric kidney. However, they degenerate in the female after formation of the metanephric kidney. In the absence of androgen, the external genitalia develop into female phenotype. A proliferation of the mesoderm around the cloaca membrane because the underlying ectoderm to rise up so that three structures are visible externally. These are the phallus, the urogenital folds, and the labioscrotal swelling. The phallus forms the clitoris, the urogenital folds form the labia minora, and the labioscrotal swelling form the labia majora and the mons pubis.
Late nights in the library, books piled high, flashcards and textbooks, we're reaching for the sky. Anatomy and physiology, it's a constant grind, but we're pushing through the tears, we're gonna make it in time. Yeah, we cry every day, but we won't lose faith. With every sacrifice we'll find a better way.